Have you ever really stopped to think about the miracle of sight? I mean, really think about it. A single photon, this massless little particle of light, travels at an incredible speed, hits your eye, and then, in just a fraction of a second, your brain builds this entire detailed conscious experience of the world. Well, today, we're going to trace that exact journey, from that first flicker of light all the way to the image you're perceiving in your mind right now.
So here's our roadmap. We'll start where the light is captured in the retina, follow the signal to a major intersection in the brain, see how it gets sorted at a thalamic relay station, and then watch it project to the visual cortex where we finally make sense of it all. Oh, and we'll even take a quick look at a super-fast reflex that happens before you can even think.
Everything you see, everything you perceive, it's all constructed right here, inside this amazing, complex organ. But believe it or not, the story doesn't actually start in the brain. It starts in your eye.
Okay, let's get into it. We're starting our journey right at the beginning of the central visual pathway, that exact moment when light is first captured by the retina.
So our journey begins in the retina. Think of it as this light-sensitive screen at the very back of your eye. This is ground zero. It's where the physical energy of light gets translated or transduced into the electrical language that your brain can actually understand.
The real workhorses doing this translation are specialized neurons called photoreceptors. You've got over 100 million of these tiny cells in each eye, and they come in two main flavors, rods and cones. Rods? Well, they're your night vision specialists. They are incredibly sensitive to even the dimmest light, but they don't see in color. They're mostly out in your peripheral vision. Cones, on the other hand, love bright light. They're what give us our sharp, high-definition color vision, and they are packed super tightly into the very center of your retina, an area called the fovea. That's exactly why you look directly at something to see it clearly. Okay, now this part is wild, because the mechanism here is completely the opposite of what you'd expect. In total darkness, your photoreceptors are actually on. They're constantly releasing a neurotransmitter that tells the next neuron to calm down, to stay inhibited. But when a photon of light hits a rhodopsin molecule, it kicks off this whole chemical cascade that reduces a substance called cGMP. This drop in cGMP closes some channels, which hyperpolarizes the cell, basically turning it off. So by turning off that inhibitory signal, it allows the next neuron in the chain to fire. So get this, light creates a signal by stopping another signal. Mind-bending, right?
So once that signal is generated, it gets passed along, and the long axons from the retinal ganglion cells, these are our second-order neurons, they all bundle together to form the optic nerve. This nerve is like a massive data cable carrying all the visual info from one eye, and it's headed straight for a major highway interchange deep in the brain.
And that interchange is called the optic chiasma. It's this cool X-shaped structure where the two optic nerves meet. And this is where something really clever happens, a partial crossover, or what we call a decussation of all those nerve fibers.
Let's break that down. The fibers that come from the inner half of each retina, the part closer to your nose, they cross over to the opposite side of the brain. But the fibers from the outer half, they stay on the same side. The beautiful result of this sorting is that everything you see in your right visual field gets processed by your left hemisphere, and everything in your left visual field gets processed by your right. It's an incredibly efficient system. And you know, this specific wiring isn't just some neat anatomical fact. It has major clinical importance. Your pituitary gland sits right underneath the optic chiasma, so if a tumor starts to grow there, it can press up against those crossing nasal fibers from both eyes. And since those fibers handle your peripheral vision, you guessed it, you start to lose your peripheral sight, leading to what's commonly known as tunnel vision. Okay, so after that big crossover, the newly sorted signals travel along what we now call the optic tracts. Their next stop is a critical relay station deep inside the brain, the lateral geniculate nucleus, or LGN, which is part of the thalamus. And this isn't just a simple pit stop. It's a highly organized processing center, kind of like a central mail sorting facility. The LGN is made of 6 distinct layers, and it keeps sorting information. Layers 1 and 2, the magnocellular layers, are all about the big picture. They specialize in processing movement and contrast. Think where and how fast. Then you have layers 3 through 6, the parvocellular layers. These are your detail-oriented layers, handling all the fine-grained information about color and form. Think what? This separation is key for processing everything in parallel.
And the organization gets even more meticulous. The LGN also keeps the information from each eye completely separate.
Layers 1, 4, and 6 get their info from the opposite eye, the contralateral eye. Meanwhile, layers 2, 3, and 5 get their info from the same side eye, the ipsilateral eye. So even this deep in the brain, the signals are still neatly filed by both their function and which eye they came from. All right, from the LGN, our visual signal is finally ready for the last leg of its journey. The neurons here, our third-order neurons, send their axons fanning out in this huge projection called the optic radiations, heading for the very back of the brain.
This massive tract of fibers, also known as the geniculocalcarine tract, perfectly preserves that spatial map of the world from the retina. Fibers from the upper part of the retina, which see your lower field of view, travel high up. Fibers from the lower retina, seeing your upper field of view, take this interesting detour forward into the temporal lobe, forming a little hook called Meyer's loop, before sweeping back. And then there's a huge intermediate division dedicated to carrying all that high-res data from the macula, your central vision.
And this brings us to a truly mind-blowing concept called cortical magnification. That tiny little pit in the center of your retina, the fovea, which is responsible for all your sharp, detailed vision, well, it gets a ridiculously huge amount of real estate in the visual cortex. Your brain dedicates an absolutely massive amount of processing power just to the very center of your gaze. So now the signals have arrived. They've reached their final destination, the primary visual cortex at the back of your head. But this is where the real magic happens. This is where we go from just detecting lines and edges to actually recognizing a face or reading a sentence.
Think of the primary visual cortex, or V1, as the first analysis hub. It gets the raw data from the LGN and starts breaking it down into basic components, like the orientation of lines and edges. From there, the information gets passed on to higher-level areas, like V3 and V4. It's in these areas that the brain really starts putting the puzzle together, identifying what an object is, figuring out where it is, and perceiving its color and motion.
But before we wrap up, let's take a quick look at a really cool visual pathway that completely bypasses all of this high-level cortical stuff. It's a lightning-fast, automatic reflex that protects your eyes every single day.
This is the pupillary light reflex. When a bright light shines in your eye, the signal travels up the optic nerve, but instead of going to the LGN for processing, it takes a shortcut to a spot in the midbrain called the pretectal area. Now here's the key. This area sends signals to a nucleus called the Edinger-Westphal nucleus on both sides of the brain. From there, the command goes out along the oculomotor nerve to the muscles that constrict your pupil. And because of that bilateral connection in the midbrain, shining a light in just one eye makes both of your pupils get smaller at the same time. This whole circuit is completely subconscious and incredibly fast. It's a pure protective reflex. Its only job is to shield those delicate photoreceptors in your retina from getting blasted by too much light.
So as we've seen, this whole journey from a photon to a perception isn't just a random scramble of signals. Not at all. It's a precise, highly organized system that maintains a faithful map of the visual world every single step of the way, from the retina to the LGN, all the way to the cortex.
And that precision is why the system can seem so fragile. For instance, a tiny little stroke affecting that Meyer's loop we talked about can knock out exactly the upper quarter of your vision on the opposite side. But this fragility is also a powerful testament to the brain's incredible organization. So what does it really tell us about the brain's precision, that it doesn't just vaguely see the world, but instead it meticulously maps every single point in space? Something to think about.